Like literally, I am not in that mood to teach. But anyways, we will discuss and we will study something. So we have already completed this part yesterday, that how our gut microbiome is associated with different neurological disorder and autoimmune disorder. Like what are the different composition of bacterial group whose concentration is increased, and what are the different bacterial group whose concentration is decreased. So we have already studied, like there are different kind of neurological disorder ranging from autism spectrum disorder to normal depression and anxiety. So these are the most common one, but they are also fatal for our health, because they are also capable of initiating inflammatory response in our body. So anything which initiates inflammatory response in our body is harmful for us. So how these neurological disorders are occurring in our body, like what are the exact pathway uh, which is influenced by the release of inflammatory mediators. So whenever an inflammatory mediators are released in the body, so where they attack, so they can attack the vagus nerve. So this one is the very common and dominating pathway, vagus nerve. So its role in neurological disorder is in the direct signaling from gut to brain. So vagus nerve is involved in the direct signaling from gut to brain. So it sends the signal from all of the body. Who send the signal? Our bacteria residing in our gut. So they help to transmit the signal directly to the brain. So this pathway is hindered if you are having the disturbed gut microbiome. Now, apart from this vagus nerve thing, you also have an modulated immune system in a negative way, like microbiota regulation, inflammation via cytokines, and microglial activation. Microglials are brain cells. So you can imagine that if you are releasing inflammatory mediators, they can attack the immune system also. They will release the cytokines like interleukin 1, interleukin 6, TNF alpha. So all these will be directly in your bloodstream, and they will start initiating the more such inflammatory responses. Now the another one and the most common one is by the synthesis of microbial metabolites. So example, short chain fatty acid, tryptophan metabolites, which affects the central nervous system health. So anything which is synthesized, but it not in a coordinated way or in a very altered way, so it will influence your overall physiology of the body. Now apart from this, you also have a neurotransmitter modulation. Whatever neurotransmitter is releasing in your body, it will be also altered, and neurotransmitters are the messengers like acetylcholine. So whenever you we use mobile phone to send message, or we also use normal phone calls. We have also social sites, social platforms like WhatsApp, we have LinkedIn, we have Telegram, we have Twitter, we have so many things. So likewise, our central nervous system also have certain things through which they communicate with each other. So the first one is neurotransmitter. So the way we message to somebody else residing in some other location other than us. So what we do, we send message. Whatever we want to convey, we send it through message, call, or any other WhatsApp chat or any other platform. So likewise, if our one neuron wants to communicate with the second one, like if this is a neuron, then these are the dendrites. So if this neuron wants to communicate with the another one, so how it will communicate? It will release some molecules, some messengers, which are known as the neurotransmitter. So if your gut microbiome is altered, or if you are suffering from dysbiosis, then this neurotransmitter will be blocked. It will not be synthesized, or else what can happen? It will be synthesized, but it will not be sensed by the another neuron, another neighboring neuron. So this way the communication is hindered, because one is producing and the another one is not receiving it, not responding towards it. So this way it is altered. Now hypothalamus pituitary axis is also influenced if you are having dysbiosis. So whenever your HPA axis is altered or influenced, then you will not have the required hormones. The hormones will not be synthesized, the required hormones. Like anterior pituitary will not synthesize the hormones, and posterior pituitary and other also hormones will be blocked. And if hormones are not synthesized, like I told you that neurotransmitter is a one factor which controls the overall signaling between the neurons. So likewise, we have hormones also, which are also a chemical messengers which are released in trace amount by different glands in our body, different endocrine glands in our body. So this will also be influenced if you are having a dysbiosis. So all these pathways are responsible for different neurological complications, different neurological disorders, or you can say that your dysbiosis or your bad bacteria will adapt all these methods to influence your normal physiological mechanism going on in your body. They can alter your vagus nerve signaling, they can modulate the immune system, or else they can block the messengers in your body. Like they can block the synthesis of hormones, they can block the synthesis of neurotransmitters, and also they will influence the HPA axis. So anything can take place. Now we will focus on the cancer, like how the gut microbiome is related to cancer. This is the last topic of this module. So how cancer occur whenever we are having dysbiosis? If our gut microbiome is altered, so we can say we can have colorectal cancer, we can have gastric cancer, we can have pancreatic cancer, liver cancer, and breast cancer. Not only just these cancers, but these cancers are the most commonly occurring cancer when we are having dysbiosis. So these are the list, like CRC, gastric cancer, pancreatic cancer, liver cancer is the very common one. And out of all these are the most common one, because they are directly influenced. If your gut is not having a healthy bacteria or a healthy composition of good bacteria, then you will be surely having some metabolic disease, and all will be influenced. So how you can have colorectal cancer? In colorectal cancer, you can expect the increase in the population of Fusobacterium nucleatum. Fusobacterium concentration will be increased, and also you can expect the production of genotoxin. Like when a bacteria synthesize a toxin which directly influence the host metabolic machinery. So this way also they alter the overall immunity of that particular host, because if our immunity is not working properly, then your immune system will not be able to recognize that this particular genotoxin or whatever they are releasing, whatever metabolite they are releasing, your immunity will not be able to recognize it as non-self. And if they are not this is the adaptive mechanism. Like this question is asked, how the antigen take entry into the host? So the antigens are smart, they try to pull down the response of the host by mimicking the self condition. Like they replicate the conditions of our own body cell, so that our immune system will not be able to recognize that this is not ours, this is not an foreign cell, this is our body's own cell. So the antigen try to make or make their appearance like our own cell. So in this way they can invade our first line of defense, they can invade our immune system. Our immune system will not be able to focus on the non-self antigen. So in gastric cancer also, the Helicobacter pylori infection increases, and this is a class 1 carcinogen. So you should know about the different kind of cancer which can be occur through the dysbiosis, and what are the different kind of bacteria which increases, what are the different kind of bacteria which decreases. So you can see gastric cancer is one of the most common cancer. If you are taking unhealthy diet for so longer, so ultimately you are destroying the natural gut composition, natural bacterial population of your gut, and ultimately your inflammation level will increase and you will be having some gastrointestinal disease. And if it is for a prolonged years, then you can have a cancer kind of thing. So class 1 carcinogens, it it is acting as a class 1 carcinogen. Class 1 carcinogens are actually, there are some categories given by the IARC. So IARC is International Agency for Research on Cancer. So it controls the level of pollutant or the factor which can be regarded as class 1, class 2, class 2A, class 2B. So these are the substances, you can say. And if you are talking about the microorganism, you can say these are the bacteria which are considered as a carcinogenic to the humans with sufficient evidence. Like this has sufficient evidence of human trials that this definitely will cause colorectal cancer, gastric cancer, whatever. So this is a class 1 carcinogen. Class 1 carcinogen means it is categorized by the IARC, that it has been known that this will definitely cause cancer with the proof, like it is proved by different research. Now pancreatic cancer. So as our gut pancreas axis is also there, so it can influence your oral gut dysbiosis, which is associated with increased risk of Porphyromonas gingivalis implicated. So this can also influence your whole body. Pancreatic cancer is also nowadays one of the most common cancer. So if oral or gut dysbiosis is increased, then you can have Porphyromonas gingivalis, and you can expect the damage of your all pancreatic cells. Now liver cancer, hepatocarcinoma. So gut liver axis is also a normal function of the gut microbiome if it is not having dysbiosis. So if you are having dysbiosis, then you will have increased gut permeability, then it will release endotoxin and then hepatic inflammation. Now leaky gut condition. So in breast cancer also, you can expect microbiome modulates the estrogen metabolism, influencing the hormone sensitive cancer risk. As in breast cancer, you can expect if estrogen levels are increased, then you can expect the breast cancer. So estrogen is a secondary, it is a hormone which control the secondary sexual characteristics of a female. So whenever you are exposed to estrogen for a longer time, like if we are having a very late menopause, or a very in early age, like in a very early teenage, we are having the menstruation cycle, so we are exposing more to estrogen and we are having a more risk to estrogen. So the more you are exposed to estrogen, the more is the chances that you will be having a breast cancer, because these modulates the overall signaling cascade of our body. So nowadays breast cancer, or also you, if you are for a very longer time, if you are taking hormonal therapies, like if you are taking some contraceptive for a longer time, because they are the combination of estrogen and progesterone, then also you are susceptible for this breast cancer. Because if your body is exposed for so longer to this secondary sexual hormone, then your body will consider it as a cells, and it will start doing the signaling cascade, and it will alter the overall cell proliferation part in your body. It will rapidly proliferate your cell, and cell division will not stop, because we have to stop our cells division at a certain point. So in cancer, what happens in cancer, we have unregulated cell division. So this way breast cancer can also take place, and this is one of the most common cancer in females. I think this is the second most common cancer nowadays since last decade observed in females. And also there are 4 variants of breast cancer, and if we are having the triple negative, then the chances are very low to get cured completely. So these are the different kind of cancers, and all these things are for a chronic period of time. They are not for a single day. If your gut bacteria is not showing a good composition for a single day, if you are having some junk food and you are having upset stomach, then you can expect a disease. No, this is for a longer period of time. These are all chronic disease, not a single day disease. So these are the different kind of cancer. Most common cancers I have listed out, which you can expect if you are having dysbiosis for over a very prolonged years, like 6 years, 7 years, 8 years, 10 years, likewise. And if you are having some other secondary complication, like if you are having diabetes, if you are having thyroid, and also if you are having high blood pressure, then all these things will be a worsen at it best. So these are the things which are associated with the module 3. Now we are done with the module 3, but also we have to focus some more about the cancer wala part, because this is important. So this one. So cancer types linked to gut microbiome. Like what are the different types of cancer you can expect if you are having altered gut microbiome or if you are having a dysbiosis. So now the question comes that if our gut microbiome is altered, then a term known as dysbiosis. So dysbiosis is what? Altered gut microbiome. Now a question arises here that how our gut microbial population or how our gut microbiome protect us in having different kind of cancer. Like it is acting as a tumor suppressor, or how it prevents the oncogenesis. So, and you can expect that tumor suppressor gene, TSGs, are known as cell break. Like whenever we are having some problem while driving a car or a two-wheeler, then what we do? We just start pulling the brake. So this is the reason that it acts as a cell break. It will stop uh, the unwanted condition. Like if the cell is rapidly dividing from 1 to 2, it is fine. From 2 to 4, then also it is fine. And from 4 to 8, then also fine. 8 to 16, 16 to 32, 32 to 64, 128 to, this is normal. But if your one cell is dividing rapidly into multiple cells, and then an overgrowth occurs, and this will take a form of known as tumor. Tumor is nothing but an overgrowth of rapid growth of cell which looks like this lesion, mass, and this is known as tumor. So this is the result of what? The failure of regulated cell division, because our body has an excellent cell division mechanism. Cell division mechanism is regulated. Like we have to go from G1 to S phase, then G2 phase, then M phase, and everything is in coordinated manner. So we also have some checkpoints, surveillance mechanism. Like when we cross one district, or when we travel interstate, then we are having checkpoints. Like people used to check that whether the things are going on a appropriate way or not, whether something fishy or not, so they check. Likewise, our cell division, we also have some checkpoints, or these are known as the surveillance mechanism, that whether the cell division is going on appropriately or not, whether one cell is dividing into only 2 cells or not. So likewise, if our surveillance mechanism fails, like if our checkpoints are altered, if they are not working properly, then this thing will happen. Not this regulated division, but an unregulated division will happen. So our gut microbiome protects us by stimulating the role of tumor suppressor genes, because we are having naturally tumor suppressor genes in our body. So when there is a mutation in this tumor suppressor gene, we can expect cancer. So, and also we are having oncogenes, proto-oncogenes, then they are converted into oncogenes. So these oncogenes are what? These oncogenes are already there in our body, but we provide the conditions which require to make them oncogenic. We are we are having proto-oncogenic genes. So if you are providing any condition which is favorable for this conversion, proto-oncogenic into oncogenic, if you are providing UV light, if you are providing some cancerous agent, chemicals, hazardous chemical, so, or dysbiosis. So these gene will be converted into what? These gene will be converted into oncogenes. So this conversion is stopped or checked by this tumor suppressor gene. So if we are having mutation in the tumor suppressor gene, then they will not protect our body, and consequently, what will happen? Unregulated cell division, then rapidly a tumor will form. So this is the reason why there are still no such therapeutic options to cure cancer at like 100%, because the time required for the tumor to grow is very less. Like it is very rapid. No antibiotics can control that, and it spreads at a very highest rate. So it is spreading all over the neighboring cell. It is making oncogenic to each and every cell residing nearby to it. So this is very fatal. So, and that too in we are having solid cancers, and we are having liquid cancer like blood cancer. So blood cancer is very fatal because your this cancerous cell is traveling across your whole body, so it will damage every cell nearby to it. So this is why blood cancers are very fatal. So ultimately, how our gut microbiome, like how our gut microbiome, these are the good bacteria, are helping our tumor suppressor gene to function properly, to don't support this conversion like proto-oncogene to oncogene. So possible tumor suppressive effect of the gastrointestinal microbiota. So they are involved in regulation in signaling pathways. Like if I discuss that proto-oncogene is converted into oncogene, so there are definitely so many pathways which are which takes place to do this, to make a cell cancerous or to activate the oncogenes. So definitely our gut microbiome do what? Our gut microbiome regulates those signaling pathway. They check the signaling pathway so that these conversion won't happen. And barrier function modulation. Barrier function modulation, they don't allow the inflammatory molecule to directly release into the blood, and also through this they exert an anti-inflammatory effect and anti-cancerous effect. They also provide they quench all the reactive oxygen species present in our body. And also, like nature has provided everything. If there is an defect, or if there is a disease, our body also has a defense mechanism. Like we are having the a very good DNA repair mechanism in our body. We are having different kind of repairs. Like if we are having a bulky lesion in our DNA, if you are having the UV radiation damage, if we are having the double stranded break, single stranded break, if we are having different base modification, so everything can be corrected by the DNA repair effect. So if you have studied the replication, DNA replication part, this is a very important one. I have so many videos related to DNA replication in my playlist in the channel, so we can check it out. So DNA replication is very important. So whenever the polymerase synthesizes the strand, the another one is exonuclease. So if 5' to 3' synthesis is occurring, then exonuclease do what? It is required for the proofreading activity, because the DNA polymerase add the nucleotide at a very high rate. So whenever the things are going at a very rapid stage or a very rapid rate, then there might be chances that there can be a misleading, like A binds with T, G binds with C. So if cross can happen, like A bind to C, T binds to G, then this has to be checked that whether the growing daughter strand has a correct nucleotides or not. So this is checked by exonuclease. One is synthesizing like this way, and the another one is checking that is there anything wrong going on while replicating DNA. So whenever there is an error spotted by this exonuclease, then it functions by cleaving this, and then they will incorporate the correct base, and then they will seal it through the ligase. So these things are happening in our body through the DNA repair mechanism. All these things we are having naturally in our body. So all these things are mediated by the gut microbiome. So if you are losing your gut microbiome, you are exposed to different kind of cancers. So take into consideration the gut microbiome are not only just associated with controlling your digestive part, digestion system, or your neurological system. No, they are also responsible for providing the defense mechanism like the DNA repair, which is the very, very important part of our body. Because if suppose at a very rapid rate our cell is dividing, each and every time the older cells are replaced by the new cells. So how this is happening? So this is happening through the DNA replication, because each cell has to receive the DNA, because without DNA no cell can exist. So DNA replication has to be occur in the S phase. So if DNA replication is occurring and it has so many errors, then your cell will be having the incorrect genomic material or the faulty genomic material, and faulty genomic material will synthesize faulty proteins, and then those faulty proteins will do what? They will hinder the normal physiology of your body. So this is known as the autoimmune disease. So our gut microbiome are the excellent factors which are we are having naturally in our body. So they check everything in your body, that whether we are having incorrect DNA replication part. So they check each and everything. Now, possible oncogenic effect of certain microorganism and dysbiosis. So now see, if you are altering this, everything is fine if you are taking care of your gut, normal natural gut microbiome composition. But if you are not providing the good condition for the good bacteria to increase their population, then you can expect all these things. The opposite of this box, like regulation in signaling pathway will be failed. So what you can say, aberrant signaling pathway, oxidative imbalance. Here the antioxidant activity was provided by the bacteria because they can synthesize some antioxidative enzymes, so you can quench the reactive oxygen species. So increase in the gut permeability, you can expect the, if gut permeability is increased, this condition is known as, I have written here, leaky gut. So if leaky gut is increased, everything will be bathed directly into your bloodstream. Inflammation will occur if anything is released directly into our blood. And DNA damage, because our DNA repair mechanism will be failed if you are not providing good condition for the good bacteria to grow. So this is the reason why we are deteriorating our own body through our minor mistakes. So you should think over it that your gut microorganisms are the boon for us, and not they are only fatal for our body if we are modulating their normal natural activity. They are in our body for helping us, they are our protagonist, but we are making them to act as a antagonist, because we are destroying the normal environment required for them to grow in our gut. So we are making it acidic, so why they will grow in it? You will also not prefer a condition which you don't like. So likewise, our microorganism will not grow in an environment which is not suitable, which is not favorable for them. So we are only responsible for increasing the oncogenesis in our body. So whenever there is an increased activity of tumor suppressor genes in our body, so we will not have any kind of cancer, because everybody has the proto-oncogenes in their body. So it activates only when you are providing the environmental condition. First of all, you have to avoid all those things which are related to cancer. Like you have to avoid the UV light, because UV light is directly related to, UV light do what? Mainly it lead to thymine dimer. So you have to avoid all those environmental factors, all those crucial factors which can directly increase the chances of your cancer. So you have to avoid UV light, you have to avoid the heavy chemical, harsh environmental chemicals, and you have to take good care of your gut microbial population. So if you are not maintaining all those things, then definitely your body will show a negative response towards it. So this is the things going on in your body. Now you can see here, this is a tumorous outgrowth, which is the unregulated cell division at a very rapid stage. So because cancerous cell also lose the property of contact inhibition, because if in our normal cell, if one cell is this and if one cell grow like this and another one is like this very close, they will stop growing because it has no space to grow. Like it only grow as a layer. But cancer cell fails this property. They lose the property of contact inhibition. They don't care that is anything growing on top of it, nearby to it, beside it, or whatever. So they grow at a very rapid and massive growth is there, and that is known as tumor. So nowadays, solid cancer has still options to get cured if it is detected in very early stage. But liquid cancer, like blood cancer, is still very difficult and challenging to cure, because the cell division is at a very massive rate. You cannot even expect that if you are taking anti-cancerous drug, so will they able to control that or not, because already it has crossed the stage 1, stage 2. It is very at its lethal stage, so you cannot predict. So all these things are very lethal. So you can expect all these things if you are having a dysbiosis condition. So mechanisms are genotoxin production, as I have discussed earlier, chronic inflammation, immune suppression, oncometabolites, microbiome drug interaction. So like in genotoxin production, you can expect the E. coli, colibactin, which causes DNA double-stranded break. Like if you are having an altered gut microbiome, which is the result of the different factors, you should be able to recall what are the different factors which are responsible for the gut dysbiosis. So you will have the increase in the E. coli, and E. coli genes will be altered and they will synthesize the genotoxin. Like colibactin, genotoxin means it will directly bind to the host genome. It has the capability to bind with the genome, because anything that is released has a different target site. Like this is not necessary that every product release or every metabolite release will act onto the genomic material. No, every molecule has a different target site. So E. coli, colibactin has a potential to cause, this is the normal DNA. So if it is having this break here and the break here, so then this is the double-stranded DNA break. So we need DNA damage, DNA repair mechanism to fix this. And chronic inflammation. So fusarium nucleatum activates TLR and then NF-kappa B, and then it will release pro-inflammatory cytokines. Immune suppression, they will obviously suppress your immunity, because if our immunity is fine, nothing can go wrong in our body. They are the gatekeepers, they are the armies of our body. They protect us from different pathogenic microorganisms. So they will suppress anti-tumor T cell response, because our T cell is involved in also providing the anti-tumorous effect. So if our T cell response is hindered or if it is influenced, then it will not provide the anti-tumorous reaction. Then the tumor progression will occur at a very rapid rate. Now, oncometabolites. So microbial metabolites like secondary bile acid may promote tumorigenesis, tumor promotion, tumor synthesis. So there are different metabolites released by the bacteria and microbiome drug interaction. So if you are taking a drug to control any cancer, and if you are having altered gut microbiome, then this drug will be not as responsive or as effective as a normal drug, because the structure will be modified by the microorganism, suppose, or if the target site or the active sites will be modified. Active sites will be modified. So everything can take place if you are having gut dysbiosis. So see how important are the gut microbiome population. Like they are protecting us in every possible way. So we should also provide them the condition they require to grow, because if they will grow, they will act as the army, they will act as our protective mechanism, they will ensure our healthy lifestyle, they will ensure the safety of our body. So this was all about this. Now this thing. So this is the diagrammatic part of this one. Like tumor microenvironment. How you can expect the formation of a tumor, which is nothing but a unregulated cell division. So you can have metabolite mediated interaction. Like if you are having different kind of metabolite released by the bad bacteria in your gut, they can bind to different pathways, and therefore they will initiate the tumor formation at a very high rate. Now, inflammatory pathways, they can alter their inflammatory pathways, like they can alter their T cell responses, they will influence the macrophages responses, because we know macrophages are responsible for what? For phagocytosis. Phagocytosis. They will engulf the overall whatever is released, like metabolites, cytotoxin. They can engulf them. That phenomenon is known as phagocytosis. So if your macrophages are not working properly, then who will engulf this? Antibody response. So your antibody production will also be impaired. No more antibody will be synthesized. So like whenever antigen takes place, take entry in our body, so the primary antibody which is released is IgM. So IgM is the first line of defense in our body. We are having IgGAMDE. So this is the code word for remembering whatever antibodies we are having in our body. And the IgG, IgA, IgM, IgD, IgE. So the first one is activated is IgM. So you can see, and also we can expect the tumor through the direct interaction between the different metabolic mediators released in our body. Like see, this cell cycle control is, if it failed, then you can expect the rapid cell proliferation and barrier disruption. If you have disrupt your barrier, everything will be leak out. See, everything is entering in your this bacteria is entering in your gut, and you are releasing this mediator in your bloodstream. So these four are the major contributors of the formation of tumor and hence cancer. So these are the methods that if you are increasing this of metabolite, then also it is fatal. And if you are expecting that your protective part of your immune system, that is the T cell, macrophages, antibody production, if it fails, then also you can expect the different kind of cancers. So all these are very important in protecting you, but if they fails, then they can cause different tumor formation. So now we have come to an end of the module 3. So I hope you are able to now understand each and every part of the module 3. So in fourth module, we have to study what? We have to study the therapeutic approaches, because already we have studied that how things are going wrong in our body and what are the different consequences associated with that. So we have already studied what? We have studied that how the things are going wrong. Things are going wrong means how our natural gut microbial composition is altered, or they are damaged, they are perturbed, they are disturbed. So we already know about the factors responsible for it, and factors we have studied, and consequences also we have studied. What are the different consequences? What are the different kind of disease we can expect? Like they are controlling our digestive system, they are controlling our immunological system, they are controlling our gastrointestinal system, and every neurological system, everything is under the control of the gut microbial population. Now, if we fail to maintain the required condition for those good bacteria, good microorganisms, then we can expect the dysbiosis. Now, if dysbiosis has occurred in any of the patient, then what are the therapeutic approaches we have up till now? What are the widely used therapeutic approach to rectify or to convert those damage in our body? So we can have microbiome engineering. Like microbiome engineering, if it is added engineering, then if our gut microorganism is producing something toxic, if a genome if a gene of a microorganism genome is producing something toxic, toxin production is there, so we can knock it out, we can delete that gene, we can modify that gene so that we can silence that gene. So that is known as the microbiome engineering. Whatever we are not liking in the natural wild type, natural means we call them wild type and mutated type. Wild type and mutated type. So if we are not liking any of the characteristics of the wild type microorganism, we can modify them, we can engineer them, we can manipulate its genome as per our interest. So this is known as microbiome engineering, and we thanks to the different and advanced biotech instrumentation and techniques, this is possible to alter the genome of every bacteria. Now in our lab also, I am doing this. I am trying to clone a particular gene responsible for the increased bioremediation into the wild type bacteria to increase its property towards the bioremediation. So whatever it is doing, suppose is 1x. So if you want to amplify its effect 10x, then you can clone that gene in a very hyper producer. So I'm trying to do that. I don't know it will be successful or not, but this is a very important part nowadays. Whatever you want, you can do this by your own through the genetic engineering. We have so many tools in our genetic engineering, we have so many methods, but obviously these are not easy, these are very sophisticated. They require a lot of experiments, a lot of hit and trial. Are like I have done it till more than 23 to 24 trials I've already done, but things are not going well. Like out of all these 23, 24, I have found the correct combination, like which combination is doing the best out of all the 23 and 24. So I am currently only making changes in that particular one. Like I have done 1, 2, 3, 4, so many variations. So now I am confirmed that this 13th variation is working fine with me. Now I only have to do variation in this 13. So obviously I will get something if I am very doing variation, one variation, second variation, third variation, fourth variation. So out of all these fourth variation, I may get a satisfactory, not I can say 100% good result. We can say in research satisfactory, acceptable response in this third one. So I will do again different modification in the third one, so that I can give more and more insightful comment on what are the things going on in our clone gene. So we cannot say just by doing one experiment. So this is very hectic. It requires about 12 to 18 hours to achieve this, because we have to firstly cut that particular fragment. Like we have to firstly cut the vector, then we have to insert our required gene. For inserting, we need ligase. So for ligation only, we need 7 to 8 hours. So I have to go 6 in the lab, do all the ligation reaction. Then I come at home, and after freshen up, I again go to the lab. Then I come. These things are going on. So all these things are related to the microbiome engineering. So nowadays, the cloning experiment, the genetic engineering experiment are the need of the hours. So people will ask you that whether you are having an experience in the engineering kind of thing or not. If you are applying to any abroad university or lab or any institute, because many of the lab are associated with the different metabolic engineering thing, the molecular biology labs, the cancer lab. So if you are a person who are interested in all these fields, then you definitely need to know about all these gene wala part. Microbiology is not a good thing, but after that you need to have some advanced engineering thing to, because we know that everything that is not altered at its wild stage will give an output which is obviously lower than what it will give if it is mutated. So obviously we need this mutation wala thing. So we will study that how people are doing the engineering in the wild type microbiome to achieve what we can have if it is already modified. So we will study the different kind of probiotics, prebiotics, and symbiotics. What are the mechanisms, how they act, and how they are different with each other. Like what is probiotics exactly, what is prebiotics, what is symbiotics, what are the major differences in between all these three, and what are the different benefits. And apart from that, we will study the fecal microbiota transplantation. I have used this term so many times in my previous lectures, like FMT. So this is currently in practice. Now we can have personalized nutrition. I was talking about that. If a person A has some altered microbiome, then if she or he can take only carbohydrate rich diet, only protein rich diet, only this diet, that diet, so it is not necessary that B will require the same thing. So this thing is known as the personalized nutrition, because our genome is different, our whole genetic makeup is different from each other. So what is appropriate for me may or may not be appropriate for you. So this thing is known as personalized nutrition. So as per our gut microbiome modulation, so suppose 30% modulation is there in patient A, 40% modulation, or 50%, or let's say 80% modulation is there. So there will be differences in their diet, nutrition. So obviously 30% damage is less than the 40%. So it can have some more option. A little care is required, but if you are having 40, 50, 60, then obviously the restriction will be more care has to be taken because the damage is more. Now, microbiome based drug development and future therapeutics. So we will also shed light on what are the different drugs which are in use, which are used by the dietitians or the practitioner to cure the different dysbiosis condition going on in your body, and how the microbiome respond towards that particular drug. What are the different class of drug which are currently in use? So this unit will be like the practical one. What things are going on in different medical in different medical what, like what doctors are prescribing, what dietitians are prescribing. We will study about all those things. So we only have more 5 to 6 7 minutes, so we will see little bit. So we will study about the probiotic, prebiotic, and symbiotic first, that what are the difference, exact differences between them. So see, probiotics means live beneficial microorganisms. So they are the live beneficial microorganism we are ingesting in our body. So they are mostly rich in lactobacillus. The best example is curd, because it is rich in lactic acid bacteria. So we are consuming that live bacteria, which are not at all, which are in neither way they are harmful for us. They are beneficial. They will promote the metabolic machinery boost. They will give the boost. If we are having a very lethargic digestion, very lethargic, we feel low at energy, then all these live bacteria will perform the metabolic machinery at a very rapid rate, and we will feel active, light and active. So you can see yogurt, kimchi, and different probiotic capsule which contains the live preparation of different microorganisms. So this will whenever you are having a IBS, diarrhea, or any kind of thing, your doctor will always prescribe probiotic to you, because probiotic will do what? Lactic acid bacteria will stop the liquidity of your stool, so it will not allow you to have the consequences of dehydration. It will just stop, prevent the diarrhea condition, and bowel syndrome also it will improve. And now prebiotics. So prebiotic are non-digestible food ingredients that promote growth of good microbes. So this is the difference. Probiotic means you are ingesting, you are eating the live beneficial microorganism, but in prebiotics we are eating something that will promote through different means the good microorganisms in our gut. So like this depends on our diet, and this we are taking directly the, you can say, supplement, or you can say directly we're engulfing or ingesting the microorganisms. And symbiotic, syn means combination of two. So combination of probiotics and prebiotics are known as symbiotics. So both living and non-living are known as symbiotic. So it will improve the survival and colonization of the probiotics. So we will study that lactobacillus is a part of probiotic. Inulin is a part of this prebiotic. So fortified supplement. What are the fortified means? Fortification mean increasing the natural nutrient content of the food. Like you have studied the mushroom fortification, wheat fortification. So this word fortification means to increase the whatever natural nutrients in the food item up to whatever extent we can, like 1x to 2x, 3x, up to whatever be the possible limit. So fortified foods are recommended nowadays. Like we are having organic food item like organic dal, organic rajma. So these are all pricey things than the normal one. So this will have, these are all fortified. It means they have been treated and they have been proven that this will have increased amount of the required nutrient as compared to the conventional one, the normal one, ordinary one. So your doctor will recommend to eat this wala variety and not this wala variety. So why they are saying this? Ki only try to eat the variety A instead of variety B. In fruits also we are recommended to have eat this type of fruit, this variety of fruit, and that variety of food. So all these things are related to the fortification. Whatever be the nutrient in its natural state, we are doing something, we are boosting its natural nutrient up to certain limit, then we are using it for increased nutrient in our body. So symbiotic are recommended nowadays because it will promote our gut health and it will enhance the probiotic survival. So this is the basic difference of probiotic, prebiotic, and symbiotic. So this we will continue from tomorrow. So I have already included this in detail because you should know about, because these are the basis of what we are studying. So if you have studied that what are the consequences, what is the role of gut microbial population in our body, and if you are not focusing on the therapeutic approaches, then it's no point to read all the disease and all the consequences, because we have to focus on the therapeutic approaches also. So I have included this in a very detailed prebiotic, probiotic. So what are the properties of an ideal probiotic, what we can take, what we cannot take, because we are ingesting what? Live microorganisms. So microorganisms are live, they can cause discomfort in our body. So there isn't some criteria which needs to be passed by the microorganism, because we are consuming a live thing which can modify our own immune system. So generally the bacteria which we engulf are non-pathogenic. For 100% sure they should be, they must be, not should, they must be non-pathogenic. Like GRAS means generally recognized as safe. Grass, they should be a grass category. Like if you are using Saccharomyces cerevisiae to ferment the carbohydrate to produce ethanol, they are regarded as GRAS. If you are using lactic acid bacteria, they are the GRAS, generally recognized as safe. That it has been proved that it is safe for our body. It will not cause any kind of disease. Now they should be acid and bile tolerant. So there are different properties, different criteria. So the probiotic which fulfill all those mentioned criteria will be passed for the human consumption. So there are so many different kind of clinical trials for each and every probiotic which are released into the market. So don't think that anything is released without any research. Everything has a different steps, which different protocol which needs to be followed when there is an food related item. In food microbiology, we know about the different steps which should be fulfilled appropriately and satisfactorily in order to release into the market. So we will also study this and prebiotic, obviously, and what are the ideal characteristics of a prebiotic. We will then study synbiotic. So we will then continue from tomorrow. So now we will focus on tomorrow the sport module. So stop the recording.